Niacin's potential role in protecting against Alzheimer's disease is a striking example of a scientifically grounded, biologically plausible intervention that has been largely overshadowed by trendier, better publicized nutrients like omega-3 fatty acids, curcumin, or vitamin B12. Yet, when you examine the mechanisms of Alzheimer's pathology, mitochondrial dysfunction, oxidative stress, neuroinflammation, impaired DNA repair, and NAD+ depletion, niacin emerges not as an ancillary player but as a central molecule capable of addressing multiple root causes simultaneously. Epidemiological studies, basic research, and clinical observations all point to a neuroprotective effect, but niacin remains underemphasized in cognitive health protocols, due largely to lack of commercial interest and scientific attention skewed by marketing and funding priorities. At the most fundamental level, niacin supports neuronal survival through its role in synthesizing NAD+, nicotinamide adenine dinucleotide, a molecule that fuels enzymes involved in energy metabolism, DNA repair, calcium signaling, and gene regulation. In the aging brain, especially in Alzheimer's disease, NAD+ levels plummet. This loss contributes to mitochondrial dysfunction, increased oxidative damage, impaired autophagy, the cell's ability to clean itself, and eventual synaptic and neuronal death. Boosting NAD+ is therefore a major target in experimental Alzheimer's therapies, with pharmaceutical and nutraceutical research centered on expensive compounds like NMN, nicotinamide mononucleotide, and NR, nicotinamide riboside. Yet niacin, the most accessible and proven NAD+ precursor, is largely left out of this conversation. Epidemiological data strongly suggest a protective relationship between niacin and cognitive decline. A landmark study from the Chicago Health and Aging Project found that older adults with the highest intake of dietary niacin had a significantly lower risk of developing Alzheimer's disease compared to those with the lowest intake. This effect held even after adjusting for age, education, caloric intake, and other B vitamins. It wasn't a marginal difference either. The reduction in Alzheimer's risk was around 70% in the highest quartile of niacin intake. These findings are consistent with other observational studies showing associations between higher niacin status and better cognitive function, slower cognitive decline, and reduced risk of neurodegenerative disease. Mechanistically, niacin exerts its neuroprotective effects through multiple pathways. Mitochondrial support. Niacin-derived NAD+ is essential for mitochondrial function, which is heavily impaired in Alzheimer's. Dysfunctional mitochondria generate excess reactive oxygen species, triggering inflammation and cell death. By supporting mitochondrial energy production, niacin helps keep neurons metabolically functional. DNA repair via PARPs. Neurons are especially vulnerable to DNA damage due to high oxygen consumption and limited regeneration capacity. PRP enzymes, which repair DNA breaks, consume NAD+ to function. Without niacin, NAD+ levels drop and DNA repair falters, accelerating neurodegeneration. Epigenetic regulation via sirtuins. Niacin also supports sirtuins, NAD+, dependent enzymes that regulate gene expression and promote cellular longevity. Sirtuins modulate inflammation, synaptic plasticity, and neuronal survival, key factors in preserving cognitive function. Reduced neuroinflammation. Niacin, particularly in its niacinamide form, has been shown to reduce markers of neuroinflammation, including TNF-alpha and IL-6, both of which are elevated in Alzheimer's patients. Lowering this inflammation may help preserve brain structure and slow disease progression. Enhanced autophagy. Proper turnover and recycling of cellular debris, autophagy, is impaired in Alzheimer's. NAD+ supports autophagic pathways that help neurons remove damaged proteins, including beta-amyloid plaques and tau tangles. Despite all of this, niacin has been sidelined in favor of more marketable interventions. Omega-3s and B12, while important, don't address the core metabolic breakdown seen in Alzheimer's. Meanwhile, niacin's benefits are obscured by its humble profile, its association with flushing, and a medical system that often prioritizes pharmaceuticals over foundational nutrition. Even among integrative practitioners, niacin is rarely top of mind when treating early cognitive decline, despite its powerful, system-wide relevance. Part of this neglect may be due to a simplistic view of vitamin sufficiency, the idea that if someone isn't showing overt deficiency, like pellagra, they must be getting enough. But Alzheimer's and other neurodegenerative diseases are not the result of acute deficiencies. They're the culmination of decades of marginal insufficiencies, cellular stress, and inadequate repair capacity. Supporting NAD+ levels through affordable niacin supplementation could represent a low-cost, high-reward strategy, especially when introduced in midlife or early cognitive decline. And niacin is safe. At modest doses, niacin or niacinamide, 100 to 500 mg/day, can elevate NAD+ without causing significant side effects. Even in higher doses used therapeutically, niacinamide avoids the flushing side effect of nicotinic acid and appears to be well tolerated in elderly populations. In short, the neglect of niacin in Alzheimer's prevention and treatment is more than a clinical oversight. It's a missed opportunity to address the disease at a metabolic level. We chase billion-dollar drugs and exotic interventions while ignoring the basic biochemistry of neuronal health. Niacin doesn't just support the brain, it enables it, providing the fuel and structure needed for resilience, repair, and regeneration. Until we give this quiet vitamin the attention it deserves, we risk fighting neurodegeneration with one hand tied behind our backs.